Hello everyone, I'm Damon Gannon, candidate to teach Mars 4380, Marine Fisheries Biology, in the 2024 Marine Biology Spring Semester at UGAMI. I haven't met some of you, so here is a photo to put a face with a name. I'd like to tell you a little bit about my research and teaching. I was Assistant Director at UGAMI in charge of academic programs from 2016 to 2022, and I founded the Marine Biology Spring Semester program and taught Mars 4380 twice before as part of this program, in 2020 and 2021. I also directed and co-taught the Coastal Summer Semester program at UGAMI from 2017 to 2022. I spent the majority of my career working at field stations and marine labs. I did my master's research as a guest student at Hui, and then worked as a technician there for a couple of years before doing my PhD at the Duke Marine Lab in Beaufort. I did a postdoc at Mote Marine Lab and stayed on there for several years as staff scientist and deputy program manager. While I was at Mote, I taught summer field courses at the Duke Marine Lab. Prior to coming to the UGA, I was the director of the Bowdoin Scientific Station, a remote island-based field station in the outer reaches of the Bay of Fundy that is operated by Bowdoin College. Since I left UGA in 2022, I've been sailing the east coast of the US with my wife Janet, and we've been writing about our experiences. As someone who has had teaching and administrative responsibilities at field stations, my research interests necessarily became quite diverse. Rather than following the typical process of coming up with a set of research questions and then deciding where the most appropriate location was to pursue those questions, I was already based at these field sites. And so I had to identify lines of research for my students and I to pursue at these locations. This made me a scientific generalist, which has turned out to be an asset for mentoring undergraduate students with diverse interests. My early research focused on identifying and quantifying sources of mortality for marine mammals, which often turned out to be bycatch in commercial fisheries. That led me to questions about trophic ecology and habitat selection of marine mammals, fishes, and seabirds. I soon realized that it's difficult to understand marine mammal foraging without considering bioacoustics and behavioral ecology of both marine mammals and fishes. This bioacoustics work led me to testing of less invasive sampling techniques for fishery stock assessment using passive acoustic monitoring. While studying the abundance, distribution, and acoustic behavior of prey fishes eaten by the well-studied population of bottlenose dolphins in Sarasota Bay, Florida, I experienced some scientific serendipity in the form of a red tide bloom. The massive fish kills resulting from a Carenia brevis bloom ruined a lot of the research plans that I had at that time. But I quickly realized that this was an opportunity to study the effects of Carenia habs on individual fitness, population dynamics, and community structure of fishes. The Carenia red tide blooms created a natural experiment in disturbance ecology. We had several pulses of red tide that varied in severity, spatial extent, and duration, which allowed us to see how the community responded. Here's a simple example of changes in abundance of pinfish throughout the Sarasota Bay region and adjacent Gulf waters. These are catch per unit effort data from purse seine surveys stratified by habitat type and region during a period free of red tide on the left and during a subsequent bloom on the right. A dramatic decrease in pinfish abundance in the central and southern part of the study area coincided with the bloom. Fish species vary drastically in their sensitivities to Carenia red tides. The red tides alter community structure, but the community returns to its pre-bloom conditions in usually less than 2 years. Of course, we don't know if there is a tipping point that might be reached if bloom frequency or intensity increases over time. I've taught undergraduate courses in fisheries, marine mammal biology, marine conservation biology, marine and estuarine ecology, and general biology. With respect to just field-based courses, I co-taught marine mammal biology at the Duke University Marine Lab and ecology of the Gulf of Maine and Bay of Fundy at Bowdoin College. At UGAMI, I taught or co-taught several field courses, including Special topics in ecology: ecology of the Georgia coast, which is ecology 3480. Field animal behavior, biology 3720L. Methods in marine ecology, ecology in Mars 4225. Field study and oceanography and marine methods, Mars 4500. And of course, marine fisheries biology, Mars 4380. I taught Mars 4380 during the marine biology spring semester programs in 2020 and 2021. While teaching this course at Sapelo, my approach is quite different from what it would be if I were to teach it at Athens. At Sapelo, I put a heavy emphasis on the field aspects of fisheries science. Examples of some of these activities include exercises in fish diversity, taxonomy, life histories, and growth and allometry. Here we're collecting specimens in the field. The students key them out, and of course they take a lot of photos with their cell phones. We then take a sample of fish back to the lab for the students to weigh and measure. They extract otoliths or ear bones and examine their stomach contents. They learn how the otoliths can be used to identify prey remains and stomach contents of predators and to estimate the sizes of the prey. They then use their data to construct regression equations to estimate the length and weight of fish from the length of their otoliths. The Georgia coast is home to a large number of noise-making fish, most of which form choruses during their spawning seasons. These include economically important species such as drums, sea trouts, and croaker. So we use passive acoustics to document the locations and relative sizes of spawning aggregations in the Duplin River. Students learn how to use a hydrophone, process the acoustic data, identify the species of the callers, and quantify their calling activities. Because we have been collecting these data for several years, the students can look at interannual variability. I try as much as possible to introduce students to people and agencies involved in fisheries science and fisheries management. I also think there is great value in having students contribute to ongoing research programs. The Sapelo Island National Estuarine Research Reserve and UGA Marine Extension have been conducting monthly juvenile fish surveys in the Altamaha, Doughboy, and Sapelo Sounds. So the students meet Rachel Guy, who is the research coordinator at SINER, and Brian Fleck, the associate director of MAREX, to talk about the survey's goals, their field protocols, and some of the findings to date. Then the students go out in the field and help Rachel and Brian with their sampling work. They really get excited about contributing to a real research program. I use another well-established research program as a way to teach the students about tagging studies and mark recapture methods. The U.S. Fish and Wildlife Service has a cooperative horseshoe crab tagging program that has research sites up and down the East Coast. We have partnered with SINER personnel to tag horseshoe crabs coming ashore to spawn on Sapelo Island. Horseshoe crabs are an important fishery resource as well as a keystone species. The classes in the marine biology spring semester are scheduled in 1-month blocks, with the marine fisheries biology class starting on April 1st. This schedule coincides well with the spawning season of the horseshoe crabs, so this works out quite nicely. After tagging crabs, we talk about the types of information that tagging studies can provide, such as demographic and life history parameters, movement information, habitat use patterns, and abundance estimation. We talk about the various types of tagging technologies and how they are used with a variety of species. Then we get into data analysis. We start with simple mark recapture abundance estimation using the Lincoln-Peterson estimator, paying particular attention to the assumptions of the models and what happens when you violate these assumptions. Then we use a tool called Program Mark to construct more complex Jolly-Sieber models that allow students to estimate survival, recruitment, and the rate of population change. The first time I taught Mars 4380 was in the spring of 2020 during the beginning of the COVID pandemic. The timing was perfect for teaching a fisheries field course. The 4 weeks allotted for the course began immediately after spring break, a time when the weather is absolutely ideal to be in the field on the Georgia coast. We came back from spring break and of course found out that we were going to remote teaching immediately. The rest of the university had an extra week to get ready for the change, but because of the compressed timeline of my course, I opted not to take the extra week to transition to the online format. So I had to throw away most of my plans for the course and quickly improvised a class project based on existing data that I had in hand. I had a lot of trawl data collected over the years from the Duplin River, and all the students had been out trawling earlier in the semester, so they had a basic familiarity with how the data were collected. Using the trawl data seemed to be a good option, but I wanted the students to go beyond just a simple analysis of diversity or estimation of biomass. So I asked them to use the trawl data as part of an effort to estimate the Duplin River's carrying capacity for bottlenose dolphins based on prey availability. This was a higher level application of the data with a surprisingly complex workflow that required them to use the literature, find sources of ancillary data, and make some carefully considered modeling assumptions. In this situation, the project worked well because individual students could work on pieces of the project by themselves, but they also had to collaborate because each of these pieces depended on other pieces. One of the activities that the students enjoy most and get a lot out of is role-playing in a mock fisheries management meeting. There are a multitude of stakeholder advisory committees appointed to help government agencies develop science-based fishery management plans that are compatible with how the specific commercial fisheries operate. In the 2021 spring semester, I led the class in a mock meeting of NOAA's harbor porpoise take reduction team. Take reduction teams are mandated under federal law to develop and then monitor the effectiveness of take reduction plans designed to reduce the bycatch of marine mammals to levels below a threshold called potential biological removal. Each take reduction team is tasked with working on a specific marine mammal bycatch problem in a specific fishery. Take reduction teams are comprised of fishers, scientists, managers, and conservationists. Currently, I serve as a scientific member of 3 take reduction teams, including the harbor porpoise take reduction team. So I'm intimately familiar with how these bodies operate. For the class, I posed a hypothetical problem of an increase in bycatch in the New England groundfish gillnet fishery that exceeded the potential biological removal for the Gulf of Maine stock of harbor porpoises. In this exercise, students are required to understand how potential biological removal is calculated and the data used that go into those calculations. This coming spring, I plan to have students take part in a mock meeting of the Atlantic States Marine Fisheries Commission's Atlantic Menhaden Advisory Panel. In terms of biomass landed, the Menhaden fishery is the largest fishery on the Atlantic coast, though not many people have heard of it. The question I intend to have the students grapple with is on the allocation of the catch. Given the status of the Atlantic menhaden stock relative to biological reference points used to guide the management, how large should the catch be, and how should the catch be allocated among the various sectors of the fishery? In addition, should there be a specific allocation made for predators that eat menhaden, many of which are themselves important fisheries resources? For the upcoming semester, I plan to introduce students to a population modeling tool called Vortex. Using a variety of demographic, genetic, and mortality data, Vortex can perform population viability analyses to forecast future population sizes and estimate the probability of extinction. Vortex simulates the complex dynamics of populations and how those dynamics may change through time in the presence of changing human activities such as fisheries harvests and bycatches. The model system that I plan to use is king salmon, many populations of which are in severe decline, and they are vulnerable to capture in fisheries targeting other more abundant species, such as sockeye salmon. The beauty of Vortex is that it can be used to perform sensitivity analyses for individual life history and mortality parameters, and you can simulate scenarios to predict the effects of specific management actions, such as changes in fishing effort in the sockeye fishery. Over the past 25 years or so, fisheries management has been shifting from the single species model to ecosystem-based fishery management. I plan to introduce the class to this concept through a suite of modeling tools called EcoPath with EcoSim to create a model of the Duplin River ecosystem. EcoPath is a static mass balance snapshot of the system, and EcoSim is a time dynamic simulation module used to explore the effects of specific policy changes. We'll use our existing trawl data and lots of data from the literature to parameterize the initial EcoPath model. And the great thing about this is that there are hundreds of published ecopath models in a public database that are freely available to use, including models of estuaries from the southeast US. So we can begin with one of these existing models as a starting point and modify it to better fit the Duplin River. Once the basic ecopath model is finalized, EcoSim can then be used to perform time series simulations to explore the effects of a variety of management measures, such as new quotas or size limits on certain species, to see if they achieve the desired management goals. Throughout the entire course, a major theme that I emphasize is field safety and maritime skills. My expectation is that students should come away from this course with the skills to safely and competently undertake unsupervised field research as a technician or graduate student in fisheries or marine science. After all, if they can't get to their study site, collect their data, and get home safely, it really doesn't matter how good of a scientist they are. I hold a master's license from the U.S. Coast Guard with multiple endorsements, and I have been formally and informally teaching maritime skills to undergraduates for over 25 years. I've served on about half a dozen graduate committees and supervised over 100 undergraduate research projects. These have included course-based undergraduate research experiences, independent studies, summer fellowships, REUs, senior theses at the Duke Marine Lab, Mote Marine Lab, Bowdoin College, and UGAMI. Topics range from benthic ecology, fish and fisheries, marine mammal biology, seabird biology, and even bats. The projects listed here are just a sample of some of the fisheries-related undergraduate research projects that I supervised at UGAMI. 6 of the undergrads working under my supervision published papers based on their research, 3 as first authors. Dozens of my students have gone on to graduate school and careers in fishery science and marine policy. Some examples of my recent students who I had taught at UGAMI include Danielle Jenkins, who is a current MS student in marine sciences at UNC Wilmington. Hannah Kepner, a current MS student in the School of Fisheries and Oceanography at the University of Alaska Fairbanks. Jamie Rogers on the left is a graduate student in fisheries, aquaculture, and aquatic sciences at Auburn. Brittany Pepper, a recent graduate of the Masters of Coastal Environmental Management program at Duke University. Bailey Rains on the left is working as a fisheries research technician for the Florida Fish and Wildlife Research Institute. And those of you who have been down to Sapelo recently may have met Jonah Rigdon, the fellow in the background of this photo who is driving the boat. He's a research technician working for the Georgia Coastal Ecosystems LTER at Sapelo. And I should point out that neither Bailey nor Jonah had any boating experience prior to taking my classes. Now their maritime skills are facilitating a lot of research. So that's all I wanted to talk about today. Thank you for listening. I'm really looking forward to teaching Mars 4380 this spring. It's going to be great to be able to focus on the teaching without the responsibilities that go along with directing the program. Feel free to contact me if you have any questions about any of the topics I touched on here, or if you have any questions about my plans for the spring course. Thanks, and take care.